everyone, welcome to my channel Pure Biology. Today I come with another video, that is the DNA repair, the second part. This is the excision repair. Okay, this excision repair, it involves how, okay, and another thing is that the first part of the DNA repair, that is already I have explained in my previous video, so you just check out. The excision repair involves the excision of the segment of the polynucleotide containing a damaged site and followed by resynthesis of the correct nucleotide sequence by the DNA polymerase. Simple thing is that the damaged nucleotide, that is totally replaced by the correct nucleotide with the help of which enzyme, that is the DNA polymerase. Okay, it's two parts or two types, you can say. This is the first type, base excision repair. So it initiates the removal of the damaged nucleotide base, okay, and excision of the short piece of the polynucleotide and resynthesis with the DNA polymerase. So here the base, in only one base that is already damaged or the wrong base which is not fitted with the another base, then it is replaced by the, and this total function is done by the DNA polymerase. Okay, and it is used to repair many minor damage. That means this repairing process is seen when the DNA is damaged in different way, like that alkylation or the deamination, okay, or exposure to the mutagenic agents. So this type of repairing system is seen, and DNA glycosylase here, it initiates the repair process. This is another enzyme. You have to remind that the DNA glycosylase, okay, DNA glycosylase, this initiates the repair process. And this DNA glycosylase that does not cleave the phosphodiester bond. So phosphodiester bond means the bonds between the two nucleotides. So phosphodiester bond is not broken by this. Instead, cleave the N-glycosidic bonds. Okay, I have a wrong word, that is the phosphodiester bond means the phosphate sugar bond. Okay, so here the phosphate sugar bond is not disturbed. Only which bond is disturbed? This is the N-glycosidic bonds. These are disturbed. Okay, so now what happened after just breaking of this N for nucleotide? Okay, nucleotide glycosidic bonds liberating the altered base and generating an purinic or an pyrimidinic site or AP site. So what is saying? I repeat this line again. Instead of breaking the phosphodiester bond, the N or nucleotide glycosidic bonds is just cleaved. Okay, and the site is known at that time is the AP site. AP site means apurinic site, you can say, or a pyrimidinic site. Okay, and apurinic site or an pyrimidinic site, that means without purine or without pyrimidine base. Okay, this is called the AP site, and the resulting AP site is then repaired how? By an AP endonucleases, and these enzymes introduce chain breaks and by cleaving the phosphodiester bonds at AP site. So what is happening here? In the first case, the N-glycosidic bonds, these are cleaved, but this is done by the which enzyme? This is called the DNA glycosylase. But phosphodiester bond also is broken down, but which enzyme? This enzyme is endonucleases, AP endonucleases. Okay, so its function is that just is totally the damaged base is removed from that chain and the correct base is introduced there, by which enzyme? The enzyme is DNA polymerase 1, DNA ligase and exonuclease. So 3 enzymes why needed? Because DNA polymerase 1 and exonuclease that help to extend the chain, and DNA ligase, its function is that it helps to join or attach the nucleotide bond at the proper site of the DNA helix. Okay, join, or you can call that ligase acts like the adhesive enzyme. So this is the process of DNA repairing, of excision repairing process, base excision repairing process. Simple one, the damaged base, this is replaced and the chain is resynthesized by the proper or the correct base. This is the actual process. Okay, so next part is that, or next type is that, the nucleotide excision repair. Okay, so continue with this. Okay, another thing is very important that I have to say that there is a rough diagram where I just try to explain how this base excision occurring or base excision repair is occurring. So suppose this is the U and this is G base, and U is not proper base for the G. The proper base, okay, uracil is proper base in case of RNA, not DNA. Okay, so in case of DNA, the base pair that is attached with the G, that will be C, or that must be C. So here what is happening? The uracil DNA glycosylase that activating on this proper double helix and create AP endonuclease and phosphodiester, remove the sugar phosphate. First of all, this is the single double helix structure, and where you can see that U is totally binding with the G. Okay, and after the activation of this glycosylase, that U is not remained there. Okay, so first the damaged part must be identified, and the initiation of the repairing process that is started by the glycosylase, and you can see that the total base pair is removed by the from this single double helix DNA strand. Okay, double helical DNA strand. The total base that is attached with this G is removed from this space, and then the phosphodiester bond is also removed, and by the help of the phosphodiesterase enzyme. And after that, what is happening? The DNA polymerase 1 and ligase that are activating on the single double helical strand or DNA double helical strand. Then the actual base, that is C, come here and attached with the G. C comes here and attached with the G. So in this way, damaged base is replaced by the correct base. Okay. Second part of the DNA excision repair, that is the nucleotide excision repair, I will give the explanation in my next video. So continue with my channel.